genome sequencing. And that comes to a lot of your commercial labs that many of you may be familiar with. So when we're talking about whole exome sequencing, we're basically looking at analyzing DNA and RNA that are basically with whole genome sequencing. I'll just start there. That's a bigger picture. Yeah, it's a lot harder to do because you're looking at everything that's going to be made or not be made. Whereas whole exome sequencing, it's a lot more targeted.
So a lot of your DNA is what we call junk DNA.
Yeah.
It's there and does
it,
nothing. And so whole genome is you sequence everything, including the junk.
All the junk.
Where whole exome is saying we're just going to sequence the stuff that we know actually has a biological purpose.
Exactly.
Yeah, and that's the difference. And the importance of that goes back to what we discussed before. The more things you look at, the harder it is to know what's there. And so trying to focus on the non-junk we think is advantageous.
Exactly. And again with hotspot panels. So we always say 10 years ago, there were maybe 50 or 100 genes that were actually relevant in cancer that we needed to know about. Then it was 300 genes. So when we started, we called them hotspot